ಇಲ್ಲಿ ಗ್ಯಾಪ್ ತೆಗಿ ಲೈವ್ ಇದೆ ಕಂಟಿನ್ಯೂಷನ್ ಬರ್ತು ಸ್ಟಾರ್ಟ್ ಮಾಡೀನಿ ಈಗ ಹೊಂಟದ ಲೈವ್ ಅಪ್ಲೋಡಿಂಗ್ ಲೈವ್ ತೋರಿಸ್ಬೇಕು ಲೇಟೆಸ್ಟ್ ವರ್ಟಿಕಲ್ ವರ್ಟಿಕಲ್ ಇದೆ ಈಗ ವರ್ಟಿಕಲ್ ಕರೆಕ್ಟ್ ಅದಲ್ಲ ರೈಟ್ ವರ್ಟಿಕಲ್ ಇದೆ He has been a guide for 6 PhD students and more than 100 research interns, each for about 6 months. He has experience with more than 6 Indian universities and institutes, including Banaras University, where he completed his PhD in 1996. While most of his studies were in India, he also has academic experience in countries such as USA, where he stayed for 3.5 years, 3.5 years, and Australia, South Korea, Conference Talk, and experience and interpretations. He has served as a head of production at Packet startup in Mumbai for founded and served as the director of Shodhaka Life Sciences Private Limited for 10 years, but continuing to do justice to the main responsibilities at IBAB. Currently, he is only a scientific advisor to Shodhaka. He founded a virtual data and analyzer organization that is hosting an international bioinformatics resource guide to biologists. His education, he has completed his BSc PPC in Udupi, MSc in Zoology from Gulbarga University, before joining a research project at Mangalore University, and then moving to Banaras University for his doctoral work on mammalian reproductive pheromones. He conducted postdoctoral research at IISc Bangalore and University of Virginia, USA. His teaching and research programs, he taught multiple subjects including bioinformatics, data mining and analysis, molecular biology, genomics, transcriptomics, proteomics, biostatistics and research methodology at IBA and elsewhere. He has been a adjunct professor at University of Sydney, Manipal Higher Academy of Higher Education, JNCSR, IITB and CHG. He has been an invited speaker at more than 50 national, institutional and international conferences, workshops, as well as about 100 other local events at various academic and industrial organizations. Apart from regular teaching, he has designed and organized more than 50 short courses and workshops for students, teachers, scientists, and other professionals and organizations. He also conceptualized and convened a unique, for about 14 months, laboratory biotechnics program at IBA through 8 successful batches, which witnessed a placement of 95% in every batch. He was also among the first in India to conduct more than a dozen 10 days hands-on omics training modules after the advent of NGS. He is passionate about imparting knowledge and about planning and preparing for higher studies, jobs and careers, and frequently conducts online educational programs for youngsters. With this brief introduction, I would like to welcome Shridish K. Acharya for this National Conference on Multiomics and PIA Web for Understanding Gene Expression Regulation. I welcome you, sir, on the dais. Please give a big round of applause to Shridish Acharya for the conference.
Thank you very much, sir, and thank you very much to the organizers. It's a pleasure to be at a place where you have done your masters. So, currently, ನಾನು MSc ಮಾಡಿ zoology department.
ಸ್ವಲ್ಪ ಆಸೆ
It's very nice actually. I think I've attended a couple of workshops, conferences after coming back from USA at Gulbarga University, and they were mostly organized as zoology department. After so many years, I was so happy to hear one of the invitation. It's really a pleasure. So for the conclusion today, I'm 1 hour talk for half an hour. ಆಮೇಲೆ half an hour I'll try to keep it short. ಉದ್ದೇಶ ನಂದು ಭಾಷಣ ಮಾಡೋದಲ್ಲ. Okay, so ಉದ್ದೇಶ ನಂದು ನಿಮ್ಮ ಜೊತೆ ಮಾತಾಡೋದು. So I would like to talk to you all. Okay, so feel free to ask questions anytime in the middle of the talk. Okay, because I don't enjoy giving a lecture. Okay? I would like to talk to you. So though I have prepared a little longish talk, but I will try to find you it. So the topic is suggested, indicated here, multi-omics and AI for understanding gene expression regulation. So today what I decided to talk is about these main topics. One main research question we have. Second, what research we have done so far. What is the current research we are doing now and what we would like to do later. So what we would like to do later is about multi-omics and so why genes are expressed differently. This is the main question in our research. The whole world is, many scientists in the world are trying to address this question. We are one of them. Okay? So research so far. So expression databases and meta-analysis methods, this is what we have done so far. We have developed new computational methods for meta-analysis. So then we go look into zoology, botany, biotechnology, ಯಾವುದೇ course ಮಾಡಿದ್ರು things keep changing. So don't think that your specialty is this and that's seen forever. So I'm a good example. After my MSc, I have done actually the first thing I did is a wildlife research. I went to Western Ghats Forest and then did some studies there. And after that, I have done behavioral studies in animals. I was a molecular biologist. So that's how in US I did cloning and expression of proteins, genes, and characterize of proteins. So I'm a biotechnologist, I live in a laboratory. ಒಂದು ವರ್ಷ biotechnology company ಕೆಲಸ ಮಾಡಿದೆ ಅದು ಮುಂಬೈಯಲ್ಲಿ. ಅದರ ನಂತರ ಐಡಿಯಲ್ಲಿ as a biotechnologist. ಬಟ್ ನನಗೆ ಬಯೋಟೆಕ್ನಾಲಜಿ ಮಾಡೋದಕ್ಕೆ ಬೇಕಾದಂತ ಫಂಡಿಂಗ್ ಇರಲಿಲ್ಲ ಅಲ್ಲಿ ಹೊಸ ಇನ್ಸ್ಟಿಟ್ಯೂಟ್ ಅಲ್ಲಿ. ಬಟ್ ಸ್ಟಿಲ್ ಏನು ಮಾಡಬೇಕಲ್ಲ. ಸೊ ಐ ಸ್ಟಾರ್ಟೆಡ್ ಗೆಟಿಂಗ್ ಇನ್ ಟು ಬಯೋಇನ್ಫಾರ್ಮ್ಯಾಟಿಕ್ಸ್ ಅಂಡ್ I was good at it. So I have developed new software, new databases and new algorithms. Meta-analysis is really not a head. So regulatory mechanisms is what some insights we got. We actually found some things. On the go, we also have developed a new diagnostic assay. That's an interesting story. Of a PhD student, very young PhD student. By the time he completed his PhD, he got a research grant of 50 lakh rupees because he developed an assay which can be commercialized. Okay? So that story also I intend to tell. Let's see how much we can talk about. So currently we are doing multi-omics research. We are using multi-omics research. We have just started. We also intend to use AI approaches in analyzing the multi-omics data, and we would like to integrate multiple types of data to understand gene expression regulation. In future, we would like to understand the gene expression regulation at different levels, as you can see, and we want to take what is called precision diagnostics. Precision diagnostics are there. For example, you want to detect lung cancer. So there are some tests available, but our research has shown already that when you try to identify markers, it may not be same for everybody. We have found using the public data only that men and women have different type of markers. Okay? So you get more and more precise. Indian population might have a little different type of markers. So we are finding this type of things. Now, this is my role. I will talk about this, but what is your role? It's a question for you. Your role, students are talking to, your role is to be interactive, okay? ಏನೇ ಪ್ರಶ್ನೆ ಇದ್ರು ದಯವಿಟ್ಟು ಹೆಸಿಟೇಷನ್ ಮಾಡಬಾರ್ದು. ಓಕೆ, ನಿಮ್ಗೆ ಏನು ಗೊತ್ತಿಲ್ಲದೆ ಬಿಟ್ಟು ಅಥವಾ ನಿಂತು ಯು ಹ್ಯಾವ್ ಟು ಆಸ್ಕ್. ಓಕೆ, ಐ ವಾಂಟ್ ಟು ಟಾಕ್ ಟು ಯು. ನಾನು ಬೆಂಗಳೂರಿಂದ ಬಂದಿರೋ ಉದ್ದೇಶ ಅದೇ, ಭಾಳ ಖುಷಿ ಆಯ್ತು ಬೆಂಗಳೂರಿಗೆ ವಾಪಸ್ ಹೋಗ್ತಾ ಇದ್ದೀನಿ ಅಂತ ಹೇಳಿ. ಬಟ್ I don't want you to simply sit back and relax. Because relax ಮಾಡಿದ್ರೆ ಹೋಗ್ಬಿಡುತ್ತೆ. So don't relax too much. Try to be active, try to talk to me, okay? This session should be useful to you. All of your teachers here have taken so much trouble to arrange this workshop. Let's make best use of it. All right? Yes? So let's start with the first one, but I want to start with basics. This is always important because a lot of times we miss something. Basics, fundamentals, pillars in the building it up. So this fundamental concept clarity is very important. So let me quickly go through it. I don't mind if I don't talk about my research, but basics are very important. So you know the double-stranded DNA. You have seen this. You have seen nucleotide sequence also. What does AGCT represent? You know, right? Yes. It's a silly question to ask, but I'm sure you know. But ಯಾರಾದರೂ ಹೇಳ್ತಾರೆ AGCT ಅಂತ ಹೇಳೋದು ಈ double-stranded DNA ಹೇಗಿದೆ? So is it all along here, or here, or maybe more, or is it in the center? Center. Okay, very good. So it is in the center. So normally people consider it as hydrogen bonds cannot disform. Okay, so it's a pretty small stretch here. So you have nitrogen space one here and second here. So the sequence of nitrogen spaces is what I am writing there. So that's the DNA, that's the genetic material. But inside the cell, if I have to write one small portion of the DNA, I can probably write like this. It's inserted in yes, you know. But the cell uses some part of the DNA and expresses this as RNA molecule. And that RNA molecule is eventually translocated into cytoplasm. And there, ribosome acts on it, and an amino acid sequence produced. That's what is the functional molecule. So this is the central dogma of molecular biology we all know of, but let's go a little bit deeper. We talk about homes and omics here, genome transcription. It's the same thing. When we talk about DNA, we look at DNA to RNA to protein. We just have to remember. Okay. So we talk about DNA to RNA to proteins, but we sometimes forget that DNA is a very long molecule. How many DNA molecules are there in each chromosome? One long double-stranded molecule. Okay. So we have got 50 to 250 million base pairs in each chromosome in our body, right? So approximately like that. And all along there are some regions which are used as genes. So let's say 4 genes are there. But if I put together all the genes together, that is a genome. And if I put together all the RNA molecules, that is transcriptome. All the proteins together, then that is proteome. And that's a new terminology that has come. That is possible because today we have a different type of technology which enables us to study all molecules together. So technology has enabled us to perform large-scale studies, and that's why we are in the post-genomics era. So there are different types of like genome, transcriptome, proteome, all metabolites, metagenome we refer to, that is multiple species, DNA of multiple species together in one population, one locality. You take, for example, even if I take my saliva, I will get all different microbial species. I take DNA of all of them, all the species together. That is metagenome. Similarly, you have one epigenome, methylome, all methylation patterns. Okay, all of them together is methylome, and all the interactions between different molecules is interactome. Right? Now, when I study all those, any set of those molecules, that is referred to as omics. Okay, so genome is about DNA together. Genomics is studying all the DNA together. Similarly, transcriptomics is studying all the RNA molecules together. Proteomics is about proteins, etc. Okay. So next, so now let's go back to the concept we're talking about, gene expression. So now when I have different genes here, what happens obviously, the DNA is used by the cell. When I say, what is the first thing it is expressed as? It is expressed as RNA molecules. Okay, we get different RNA molecules, but some RNA molecules may be produced more, some may be less, etc. Eventually, they are all used to translate into proteins. So after we might have more proteins or less proteins of different types. So that proteome keeps varying across tissues and conditions. Okay. DNA is same, right? So hemoglobin I don't get in brain cells. Hemoglobin I get only in blood cells. That gene is expressed only in certain cells. Like keratin is produced only by some cells in the skin tissue, etc. So DNA is there everywhere. Genes are there everywhere, but the way the DNA is used, genes are used by the cells is different. Sometimes in the disease condition also the same tissue expresses different proteins. For example, you might have gene 3 not expressed in one condition, but under disease condition, gene 3 might be expressed, gene 4 might be shut down, and gene 2 is produced in more copies. So this way of different expression of the genes might be very much associated with the disease condition. Okay? So genes expression being different is very important, and when we look at what is happening there and how the regulation is happening, we can use these omics approaches. So we actually use all these omics approaches, and we are currently in the research that I'm going to talk about. I'm mainly going to talk about how we use transcriptomics, epigenomics, particularly metabolomics, and interactions. How do I use all of them to find out how these things are behaving in certain tissues or cells under disease conditions? Okay, that's the research we are currently doing. But I told you, overall goal is to understand the gene expression regulation. So you know the DNA, which is shown the black color here, it's a very long double-stranded DNA molecule all along. So somewhere one region is used to produce one RNA molecule. Eventually that is used to produce a protein molecule. So this is the central dogma of the shown in one context, but we are interested in the mechanism of control of gene expression. Means which gene is expressed when and where, and how many copies they are produced, and how that happens. What is the mechanism? This is our research question. So inside the cell, these things are happening. Obviously, there are different genes, and we are interested in understanding. Again, going back to basics, how gene expression is regulated. First, we have to understand what is a promoter. Okay. So you have got a gene, let's say. So a gene is, I'm marking the gene from here to there, and in that you have got promoter. So there is a transcription starts actually. Transcription starts here, and the region around it is called promoter. Okay. So this is usually around, we take plus 100, plus 200, plus meaning from the start site to here. That's plus, and this is minus. So minus 1000, minus 750 different researchers take different regions. So approximately 1000 nucleotide sequence, 1000 base pair sequence is what we take as promoter. You have got transcription factor binding sites in the promoter regions. So that is where inside the promoter you have got some really small regions, about 8 nucleotides, 10 nucleotides, 7 nucleotides, small stretches of DNA. That's where the proteins come and bind, the regulatory proteins, which are transcription factor binding sites. So that's the next thing. So promoter is there everywhere, but different for different genes. They are there in every cell, every tissue, every condition. But the transcription factors cannot be there. They are different because they are product of some of the genes. Right? So the transcription factor should come and bind and then regulate some other transcription factor might come. So eventually a few will come and bind. Then polymerase comes and binds. Then what happens is transcription begins. So then you have a final mRNA. Okay, this is how transcription is happening. So the first point is the promoter is important and the transcription factor binding sites in the promoter is important. After that, the transcription factor should come and bind. But there are other factors. So once you have got the mRNA here, this is how the transcription happens. But you could have some other types of mechanism. For example, there could be an enhancer which facilitates the promoter's function, transcription factor binding. And that might actually happen by interaction with DNA. Okay? So enhancer presence is one thing which can lead to mRNA production in a positive mode one way. Then there can be insulator. So insulator actually blocks the enhancer from interacting with some other promoters. Okay, so that is important. So when insulator is there, that interaction that was happening when it could not happen. Next. Similarly, you have methylation. So methylation of the DNA can modify promoter activity. That also happens. And then sometimes mRNA is produced, but mRNA is blocked by microRNA. That happens. Okay, so there are different levels of transcription regulation, like the gene expression regulation, like this. Then chromatin folding I talked about, right? That let me see. I'm showing here nucleosomes. You know what are nucleosomes, right? Histone complexes of DNA bound. If the DNA is very tightly packed, then transcription factor is not able to bind, it's not open. So the region should be open. So what? Then only if this enhancer will bind to interact with something like promoter or not, that depends. Or which region of the transcription factor, chromatin interacts with which other region, that changes from tissue to tissue. For example, you might have in liver cell, the gene 1 promoter interacting with one enhancer, but that region is packed, and hence that's not available, but something else might be interacting with each other. So, which same DNA, how is it packed differently in different tissues, or in the same tissue but different conditions? The way it's packed differently will affect the expression. Okay. So these are all different things that we already know. I'm only knowing the basics so far. So you can have, these are the mechanisms that I have told. What I have told is different ways that the expression can be regulated. Now, since you have not asked any question, one question to you, can you recollect all the simple methods that I told about? So I talked about, first thing I talked about is transcription factor binding site in the promoter. That will be different in different genes. But the genes and promoters are same everywhere. But transcription factor availability will change. I know that. Enhancer may be there in the neighborhood differently in the different neighborhood of different genes. The insulin can be different. Methylation pattern can be different. Most importantly, the way the protein is packed can be different. Okay, these are the things I do. Do you have any questions? Okay, since I really have to go faster, if it's your choice to ask questions.
So even external factors are also may depend on gene expression.
Yeah, so that is how the whole
Epigenetic.
what controls transcription factor expression, for example. Now, transcription factor is also a protein which is coded by some gene. So whether that is available or not, chromatin folding some proteins are involved, whether they are available or not, etc. And methylation also is happening because of some proteins, whether those proteins are active or not, etc. is what is controlled by external signals. That could be hormones, that could be growth factors, which in turn are controlled by environmental factors. Sorry? Yeah, different type of biomolecules, which in turn could be controlled by our developmental stages or environmental factors, etc. That's why the whole physiology gets connected. So this is the set of genes. Now I'm going to a different question. What we do is we, by transcriptomics, we get a set of genes which are expressed differently. For example, take lung cancer. Genes that are expressed more in lung cancer compared to normal cancer, normal lung tissue. There will be some 5 genes which are expressed more. They are expressed more, meaning environments are more in copy for those genes. There are some genes which are expressed less. Those we call them as differentially expressed genes. They are expressed together, co-expressed. They have a different pattern of expression pattern. For example, you could have gene 1, 2, 3, 4, 5, 6, like that. I put 12 genes over here, for simple illustration. All of them have a transcription start site, all of them have a promoter, etc. But maybe they have a common regulation mechanism. They are co-regulated. This is the hypothesis, and we have tested that in some cases. We have found it to be true. So, for example, you could have gene 1, 2, and you can see here. So, I think this is right. So, gene 1 and this one, they have the same transcription factor binding site, right? And some of these genes have insulator, some of them have an enhancer, some of them may be regulated at the same microRNA, and that's why they are down they are expressed less. So, like this, there might be certain common expression mechanism which is actually controlling. This is the summary of economic, right? So you can see gene 1, 4, and which are marked in red, they have the same transcription factor. DNA methylation is similar in gene 2, 3, and 12, etc. This is the idea. Okay, and this is where proteomics will help us. There are techniques available that you can explore methylation, for example, in the entire DNA. For transcription, profile, and all our biomarkers we can get in one step. We would like to do all of these type of explorations in one go. How do we find it? How do we know where is methylation, where all enhancers, where all chromatins are interaction? There are techniques to do that. So we will now try to find out what are those techniques, but more importantly, what questions we ask and what techniques we use for each question. So I will spend a few minutes to go through that now. So here you can see transcriptome. When you see, we are basically asking this question: which genes are expressed into mRNAs? That is the question we are answering by transcriptomics. Proteome: which genes are expressed into proteins? When you analyze transcription factors and transcription factor binding sites, where are these transcription factor binding sites? Which gene neighborhood? And where are the transcription factors? Which tissue or condition? Then methylose, when we see, we are asking this question, where methylations occur. Are they occurring in the gene of my interest or somewhere else in the promoter? Where is the promoter? How much? Then chromatin packing also similarly we ask this question, which chromatin regions are interacting with each other? Okay, these questions are important to be understood, and why we are asking this question is very important. I was just talking to some of your teachers, and we were debating like what makes a student better professional tomorrow is this type of understanding. Okay, understanding only definitions and things like that is okay for exam, but that is secondary to your real learning. Your understanding of the concepts and thinking about them in your own way and linking across the questions and topics. Thinking your own way and thinking across the topics is very essential. How much you do it for yourself, not to please anybody, not for the exam, how do you do it for yourself is very important. So these questions are very critical to be understood. And that's how our research began. That's how these people develop techniques. Please remember that. We as students, I'm also a student even now, okay? We all as students take pride in learning something. Oh, economics, what's the big deal? Somebody invented that. They are great. We are just trying to learn that. You can learn your own technique. You can invent your own technique if you think originally. When the question burns inside, when you have a burning desire to answer the question, you will become a researcher, a original researcher. Not somebody who wants to just publish something, etc. So that is very important. So these questions are very critical. Okay, so for example, in transcriptomics, we try to find out how many RNA molecules are produced from gene 1 or 2, etc. Okay, there is also a transcriptome. There is a different version where we address non-coding RNA molecule, meaning you sequence non-protein coding RNA molecules like microRNA, long non-coding RNA, etc. These are the other types of RNA molecules which people try to study because they regulate gene expression. Next. Hi-seq is one technology where people try to find out which chromatin regions interact. Okay, what chromatin, what promoter neighborhood, what gene neighborhood binds with what other chromatin, we can find out. So Hi-seq can answer all these type of situations. You can find out the residual by using Hi-seq technology. So this is how the heat map looks like. So you have gene 1 interacting with gene 4, you will get some sort of a map like that. So this is the schematic summary of the results. Chip sequencing is one chromatin immunoprecipitation where you find out a particular protein is binding with which all regions in the entire DNA. Particularly if you are interested in a transcription factor, that transcription factor is binding where all in the DNA. You can find out. Okay. So it will show things like that in the graph eventually, and you can understand it correctly. There is another technology called ATAC-seq where you identify open chromatin region. If a transcription factor has to bind, if a chromatin has to interact, that DNA region has to first of all open it, pack it into nucleosomes and further fibers, strong oil fibers. If that is the case, it is not functional. So what regions are open, we will study separately. That technology tells you what all regions are open. So I can use that technology to find out which promoter regions are enhancers are open. Okay, thanks. Now this is a summary of all that we discussed. In transcriptome, we try to do bulk-seq, RNA-seq. RNA-seq means we apply next generation sequencing, which is the efficient way of sequencing. It is sequencing DNA. But if we take all the RNA, convert them to cDNA, and sequence the DNA. When you realize the result, you will get a list of all the RNA molecules. That is bulk simply referring to the tissue. Tissue level, when you study that, we call that bulk transcriptome or bulk RNA sequencing. Whereas the next one, single cell RNA sequencing, is very prevalent today. That has come very fast in recent decade. So you separate cells and each cell you sequence. The RNA population is different from cell to cell. There is similarly spatial transcriptomics, another version of that RNA sequencing, but it is done after histology. You know which cell you are actually taking. There are similar, all these can be applied to non-coding RNAs. The first three are for mRNAs, the last one is for non-coding RNAs. Similarly, about protein, proteome addressed by mass spec. The questions about transcription factors you can apply by different techniques like ATAC-seq and chip-seq. Experimentally, computationally, you can predict the binding sites also, which you can prove by other techniques. Methylome, you can analyze, you can answer the question without methylation sampling. You can do that by bisulfite treatment and long NGS data, long read NGS data, then chromatic tagging. There are other techniques, but I see the main technique. Okay, so far what we have done is we have covered the fundamental concepts. Okay, more than my research, this is important. Okay, because these are understanding of these concepts help me do my own research. So if you understand this type of concepts on yourself, you can do your own research. Okay, you can start doing your own research. Of course, you need funding at all, but if you write a funding, if you do not know the fundamentals, nobody will fund you because they will talk to you before funding, right? So it's possible to get funding because you understand the concepts and you can think and find solutions, and then you start the research. So let's go into my research now. So I will go very brief, but let's see how many questions that we will get, and then we will stop it. So first one, the question itself is very important. That's the first act in research, right? Research always starts with research questions. So we are interested in knowing what genes are expressed differently, and can differential expression serve as biomarker discovery? Yes, the answer. Then we became interested in understanding the patterns, and then we want to know how it happens. What is the mechanism of gene expression regulation? Because that can help in better diagnostics. Okay? So the bigger question is how gene expression is regulated, right? So for example, you take up any disease condition that has male infertility that affects the testes, endometrial receptivity, which affects the pregnancy starting, or cancer, the beginning of how cancer begins, how can it get cancer early, or how can it identify molecules which can be used for treating the disease, etc. So can you identify relevant patterns of why using gene expression? That was one of the questions. And of course, we also asked how gene expression can be controlled in any of these conditions. An example here is lung cancer. So we have compiled a lot of data sets that are publicly available. So we compiled different study data sets and we filtered and combined the evidence by using a different computational method, which was very any of you can actually develop. As I told, the reason I told you my history is I'm from zoology and I did behavioral studies, wildlife research, and then I came into molecular biology, and then I went into bioinformatics. The reason I told that, actually it's a very long way of doing research, most of your teachers will tell you, you should specialize in one and stick to it. But the point is, anybody can do anything. So in the beginning days, you figure out what you want to do. I did not do that, but that's why I changed my field. So, but you can figure it out early. You can do any of this. So combining evidence or meta-analysis we call, using all the data sets and combining and finding out the best molecules. This is a new computational method we developed. You all can do something like that. Not exactly, but anything that you take up, you apply your mind. So using new computational methods, we identified what gene signals are more reliable among thousands of genes which are differentially expressed. Which ones is more important, more reliable, we found out. And we also analyzed promoters, we understood the gene expression regulation, but we also identified markers. So our earlier work, I'll quickly summarize now. So basically we realized the gene expression patterns, like I was showing different studies are there, in some databases they are available. But for a tissue of your interest, for a disease of your interest, sometimes it's not enough. So we actually used an experts like you who sat and compiled the data for one different pattern for one species. And that was a huge effort, and we developed it in a database, and we actually used that new method also we developed. And that fact that microarray technology is very common. But the method we developed worked well. You can see here reliability score from our database and reliability score with our own NGS experiment. This is actually showing top 10, meaning those which score very well by our method. They have better agreement with the new experiment, though the technology is different. As the score then drops, the agreement drops. Okay, that means the meta-analysis method we developed is good. So we have validated our methods, computational methods with experiments also. And this is a success story, very nice one actually. So Dr. Govind is from Hubballi, and he was, after his MSc, he took a break and then came back for PhD. But towards his end of his doctoral work, he got a national award. Among 1,000 applications, he got one award, like top 50 were identified in India. He received that award. There was no funding for that, but this biotechnology ignition grant he received, and he has not yet completed his doctoral. So what we basically did is, he actually did the RNA sequencing and like discovered biomarkers. He developed a new primer design software before that, and he developed a diagnostic assay based on the reverse transcription quantitative PCR results. which came out computational algorithm, and they identified reliable biomarkers, and what is supposed to be reliable actually worked very well. So, and he wanted to develop a kit which can actually come into market in time. So that is an example of some of the research we have done. But what we are now doing is we are trying to use multi-omics approaches using all the technology that I told so far, but I'm not getting into details. I'm more interested in your questions, but I just summarized my point in these two slides. We use multi-omics integration. Basically, we try to understand how different regulatory layers we talk about, microRNA we talk about, methylation, etc., etc. All of them together, how we analyze, we would like to perform a complementary analysis. We get an integrated regulatory network so that we can identify key players. But in doing so, we also use artificial intelligence and machine learning. Probably you already know, I think if there's a lot of artificial intelligence, but if you have questions, you can take up next please. So we are trying to do the same thing here. Machine learning is very important when you have a lot of data. It can actually start predicting things on its own. Okay? So molecular interactions at different levels it can predict. So multi-omics becomes very promising if you have a lot of data. So again, this is further what I told about changing capacity. I see how I can put together. We're going to do it manually also. We're also going to do it via AI, and we can see it in perspective. So that's the state we are. Overall, this is a summary. And but not to take so much time, I have to thank the team. Never, whatever research we do, if we do something meaningful, it's always a teamwork. So a lot of experts work for this, all the work that I have summarized here so far. I've only shown a snapshot of myself, obviously, but all my work is because of experts and some very collaborators all along. Darshan was my first PhD student, and Nidhi was also next to him. Many others are there, I can't name everybody. But that's the summary of my talk. Okay. Thank you.
Thank you so much, sir. The floor is open for discussion. Particularly youngsters, research students, and research scholars can ask the questions.
So you will be disappointing me if you don't ask questions.
So one question. Basically that like you
So students, to help you, sir, asking first question for this.
like
euchromatin and heterochromatin we are seeing in human metabolomics and even diagnostic purpose. Now some researchers are focusing on heterochromatin because it is a non-coding part of the DNA. So how far this area is now moving in India?
So initially when we talked about heterochromatin, which is a tightly packed DNA, people used to study certain tissues and then cell types. What they later discovered is there is nothing like this always packed tightly and this always moving back. So the same DNA is packed very differently across tissues. And that is why they developed this IC technology and things like that. Initially it was like usual chromatin, chromosome labeling, carrier binding and all that. So they found that this neighborhood gene, first region of the chromatin 70 and the middle region of the chromatin 70 are together here in another finished state. But same two regions are very far away. That is how they discovered. So what is packed tightly keeps changing, is very dynamic across tissues, number one. Number two, it is changing across the conditions in the same tissue. And that is regulated behind the regulation mechanisms. This is what they found, and that's why we are doing. But otherwise, what is heterochromatin, all the tissues, that's less and less.
so they are using like for medicinal purpose also they are using to identify some kind of therapeutics.
That was one diagnostic essay we had developed for my original research for basic research. When we had a new director, correct. So when the new director came and he said we should do translational research. So that's when we explored biomarker discovery, and that is one diagnostic essay we developed. That is for a type of alien fertility called non-obstructive azoospermia. So for that, what would have spent currently also there are diagnostic methods, but it will take about 2-3 months or 1 month minimum. And because histology they have to do it in analyze and things like that. Then the harmonious is there. You have step 1, step 2, step 3, then do it process. And it will cost around 3 lakhs or so. This will cost probably 7,000 and it will be done in 2 days. That was it.
It's already commercially available, sir.
No. So that's a little disappointing story. He got 50 lakh grant. He got 15 for the top entrepreneurship. So we were very interested. He was probably trying to mimic his guy, which is me. So he got 15 lakhs. He got all in the all enthusiasm. After PhD, getting small salaries, but still he managed. But the next 15 lakhs, which is supposed to get after 6 months, he did not get. And the third one also he did not get. And he stopped. Then now he has gone to USA and is doing research. He's a postdoc. That project is still waiting to be taken over by somebody else.
Can we take that, sir?
Yeah, sure.
Disaster.
So that is when we started working with lung cancer. So lung cancer current diagnostics is only one type of molecule they target. What we found is men and women are different. So there are very different set of genes which are often regulated and different molecules of cancer and that. So we could now use more precise methods. So earlier, like success rate will be 70% success on detection, and there will be a lot of false positives. How that makes it better. So it is now maybe 85% for men, 85% for women, with using different markers. So that diagnostic becomes more useful. That's what we do. But that research is still going on. That's something. Yes.
Developing AIML for each marker, is it feasible?
So AML we are trying to use right now. So when we understand the mechanism better, we identify the key players.
Yes.
When we identify the key players, that would be used as a target for research, for diagnostics, for therapeutics, or for prognostics. That's what we want to do. So for AIML, we will apply a multi-omics, which we are trying to do. We need a lot of data.
That is major.
That is where
Because the disease etiology, if you go with that, again, according to the population, a lot of variations will be there.
Correct.
So developing algorithm itself, it is a major issue in AIML.
Correct.
So-
So if you have enough data, they are able to do the job. Right now, so I did not get into the details, but you have two approaches in AIML. You can apply to a lot of data sets, or you can apply irrespective of the data set.
Yeah.
So we are doing the second one because we don't have enough data sets right now.
Yes. Because for the young generation, these are all works very much enthusiastic. So for them, it is a boon.
Yes.
Are there any questions from the audience, from students particularly?
यार आगे के बाद रिजल्ट का सवाल है.
So if there are no questions, I would like to thank Jitish Acharya sir for his wonderful, very informative, inclusive talk on multi-omics. The talk underscores the gravity of the multi-omics in coming near future research. So I thank on behalf of the university, and I also thank personal behalf of the sir. Now I pass on my chairperson of the session to conclude the work.
Good morning. Distinguished delegates, faculty members, the researchers, students, and guests. As we conclude this session, it's my privilege to express our sincere gratitude to Professor Acharya sir for delivering an outstanding talk on multi-omics and AIML for understanding gene expression regulations. This presentation was intellectually stimulating, insightful, and highly informative. Professor Acharya sir, profound understanding of the subject coupled with his ability to present complex concepts into a clear and engaging manner has greatly enriched our knowledge and broadened our perspectives. The lecture not only highlighted recent advances in in the field, but also emphasize the importance of scientific curiosity, innovation, and interdisciplinary collaboration. This multi-omics, genomics, proteomics, epigenomics, methylation, and how genes are regulated rather than studying one layer isolation. And they have focused gene expression is controlled by the multiple factors like transcription factor, non-coding RNAs, multimedia provides the data depth. And also AI/ML provides an analytical power to decode how genes are turned on off in different conditions. So the enthusiastic response from the audience and the simulated discussions that followed area testament to the relevance and the impact of the ideas shared today. Such interactions are the hallmark of a successful academic gathering and contribute significantly to the advancement of knowledge. On behalf of the organizers and all participants, I extend our heartfelt appreciation to Professor Acharya sir for sharing his expertise experience with us. I also thank all participants for their active engagement and thoughtful discussions which made this session both productive and memorable. With these words, I conclude this session and please join me in expressing our sincere appreciation to Professor Acharya sir with a warm round of applause. Thank you.
Whenever it is.
Now I request Professor Shivanas sir to participate our speaker and the chairpersons.
One last thing I would say, though you did not ask questions, your eyes spoke a lot, so you're all engaging. That is one thing I should say. Thanks for that. And I would like to congratulate and wish all the best to Professor Ceremony. And also wish best for the ahead. Thank you so much.
Request Dr. Shadagara Party, Associate Professor, Department of Zoology, NMR KV College, Bangalore, for as a co-chairperson.
क्या था इधर
Good morning, it's my privilege to introduce the second speaker of the today's conference, Dr. Lata. She is the head of the stem cell facility at the Institute for Stem Cell Science and Medicine that is located in NCS, Bangalore. Dr. Lata is a fellow E-scientist and serves as the head of the Stem Cell Facility Center Institute for Stem Cell Science and Regenerative Medicine, Princeton and Space Campus, Bangalore. She obtained her master's from University. We are very much privileged and very much happy to be here with our own one of the alumni of the University. And has built extensive expertise in stem cell biology, regenerative medicine, cell culture technologies, stem cell banking, genome editing, and research facility management. Throughout her career, she has been associated with several leading Indian research organizations, including the National Center for Biological Science, National Center for Cell Science, and Avestan, etc. And Princeton, she leads activities related to human pluripotent stem cells, induced pluripotent stem cells, organized research, disease modeling, genome engineering, and cell-based screening platforms. Her work supports major institutional initiatives focused on stem cell, organized research, women's reproductive health, and the development of quality controlled stem cell resources for biomedical research. Dr. Chungi's technical expertise includes stem cell culture and characterization, biobanking, CRISPR-based genome editing support, organoid technologies, quality control systems, and advanced research training. While a complete record of her publications, patents, grants, and awards is not currently available through institutional sources. She is recognized for her contribution to establishing and managing advanced stem cell research infrastructure and enabling cutting-edge regenerative medicine research in India. With this brief introduction, I hand over to the session, Dr. Lata. Please, ma'am, please take over.
Thank you for the kind introduction. So I'll be a little bit, we heard Dr. Acharya, he was molecular biology, OMS, OMEC, but I'm going to talk about building cell systems, building biological models through how to study your molecular biology and how it should be quality controlled, standardized. But before I start all that, I just wanted to share something. I feel very happy that there are a lot of women there because I'm a woman scientist. The fact why I'm telling this is, even when I was there were more women students, more girl students in microbiology department. I'm MSc microbiology graduate in 1999. After that I joined biotechnology department for 1 year in Tech. But now we don't see that representation at this juncture. Many of my classmates, except for 2, we have come up with that. So I would request all the women, all the girls, that do not stop. It is, we will, of course, we are the primary caregivers, but please do not stop after that. And this I can tell you from my own life story, but I think that will have to be heard. So before before you even talk, I just wanted to tell this because this is something which I get a lot of times. I've spoken in many places, and people invite me saying that you're a woman scientist. The representation is very less, and it's very true. So I think that's something which you people, all the girls sitting here, whatever your hurdles, whatever your tough decisions, just go ahead. I would put it that way. Okay? Next slide. So as I said, I'm going to speak about building biological models. So I think everybody knows what are stem cells. I don't have to introduce much about stem cells. Stem cells range from totipotent to multipotent. So totipotent stem cells are nothing but the stem cells. With them, the whole embryo has the totipotent cells. What it means to say is, when it's in the blastocyst stage, there is a ICM, that's nothing called the inner cellular mass. I think I should get a sticker. Pointer. It becomes easier. So anyway, if you see the blastocyst stage, the zygote to the blastocyst stage. In the blastocyst stage, you have the ICM, the inner cell mass, that's a few cells in between, surrounded by the protector derm. So the inner cell mass gives rise to the fetus, the whole fetus, whereas the protector derm gives rise to the extra-embryonic lineages. So what are the extra-embryonic lineages? Your placenta, your embryonic sac, and the rest of it. So where do we get? So those are the are pluripotent cells. That means to say the blastocyst, the inner cell mass along with your prefected. What's the next stage of potency of the stem cells? It's nothing but the pluripotent cells. So the pluripotent cells are nothing but the inner cell mass cells, which can give rise to all the lineages in your body. Right? Next is the multipotent cells, which also are called as the other stem cells. Means to say, how does the repair in your body happen? How does the regeneration in your body happen? You get a wound, you get a cut, and it gets healed. Right? How does it get healed? It gets healed because you have stem cells in your body. And what is the goal of stem cells? It's for repair, regeneration, and regeneration. Okay? Next. So when my talk is more focused on human pluripotent cells, largely I work on human pluripotent cells, and I've been doing that for a very long time. I did have a break in between, but yes, I've been doing it long. So what I'm talking about human pluripotent cells is nothing but the cells from the inner cell mass. We make lines, but there are two types. We call the embryonic stem cells and the induced pluripotent stem cells. So one of the embryonic stem cells is the cells which have been derived from the inner cell mass of your embryo. Right? So we make it into an embryonic stem cell line. We derive it. We put it on the dish, derive it, we. Now this endlessly will grow. So that is the embryonic stem cell line. But how do you make an induced pluripotent stem cell? 20 years back, there was a person called Professor Yamananda. What he did was, he took a somatic cell, a skin cell, and he added 4 transcription factors, and that he reprogrammed it back into a pluripotent stem cell. Have you thought of now this pluripotent stem cell has potential to become any lineage? He took a skin cell. Now that cell is back into a embryonic stem cell stage, and now you can use it for your studies. Why is it powerful? Because you know that embryos, where do you get your embryos from for your birth? You get your embryos from the IVF clinics. Correct? The excess, the surplus embryos, which parents you would have conceived and do not require. Now those surplus embryos you can use for research. So that is ethically regulated, extremely ethically regulated, and many parents do not give us because of the stigma associated with using the embryos for research. So we always had it up. But the moment induced pluripotent stem cell technology came in, the whole research boomed in this field. And recently they also celebrated 20 years because the patent has expired for Professor. So yes, now it is available. Now why, before I get into all this, I just want to tell you some few applications of using these induced pluripotent stem cells. As I said, I'm talking about biological models. How do you use these stem cells? We use these stem cells for developmental studies, for basic research. We also use it for developing cell therapies. That is the far we can go. Even for drug testing, drug-induced testing. Now this is a very classic example which is done in our institute, where what they did was they had induced pluripotent stem cell. This is from patient-specific induced pluripotent stem cell. Means to say there are certain patients with dilated cardiomyopathy with associated with obesity and sleep apnea. So it's called the DOSA phenotype. So if you look at this is a wild type, means to say this gives you the generation. That's a generation where the darkened cells, means to say the person has that variant, pathogenic variant called the KCNA variant. So what is the KCNA variant? It's nothing but where potassium channel ions are being dysregulated by the DAC pathway. It means to say, because of the potassium ion channel being dysregulated, the sarcomeric organization of the cardiomyocyte is not the normal method. So the dark ones, what I wanted to say is, has the KCNA variant, the pathogenic variant. So what they have studied is, the round ones, which is not filled ones, is the normal phenotype. So what they have done is they have made cardiomyocytes from both the normal individuals of the family and of the patient carrying the KCNA variants over several, and they found the rare pathogenic strain in the Indian population. What they did along with that was, they made the induced pluripotent stem cells for these patients. So they reprogrammed their cells, made them into pluripotent stem cells, From there, they make the cardiomyocytes of both the normal phenotype and the patient. And then after that, when they saw, you can see the P184K is a Caselia variant, that the sarcomeres were not organized well. So I don't want to get into everything. Sarcomeres are nothing but the muscle fibers of the disease. And then next was the treatment. So when they treated the patients with Caselia variant, they treated the cells, the cardiomyocytes, with simvastatin, 1 micromolar. They found that it regained the organization. Now this is a very classic example. First, you're taking it directly from the patient. This is nothing but precision medicine at its birth. You're taking the person's cells, doing the genomics, transcriptomics, everything, finding out their problems, and then doing the treatment in the lab and saying we can treat this, or finding new treatments. And simvastatin is being a very old drug in the system. Next. So we talked about 2D culture. So when you grow it in a dish, it just grows in one layer. So when you take, you can see a lot of videos when you have pluripotent cells when you go to cardiomyocytes, they start eating. So very beautiful things in the dish. But here I'm going to talk about cerebral organoids again, done at my institute. Why I'm getting you to there is the quality control part of it. So here, what they did was, again, they have studied more than 200 families with bipolar disorders, and they found that many of these patients passed on that variant to their progeny. So, as again, you can see this is a typical family with a very dense family with bipolar disorder. So, what we did was, we took the control, we made them induced pluripotent stem cell line. When I say I made it, it's a team, it's not only me. I make the pluripotent stem cell lines, they use the later. Okay? So they took the control line, controls patient, and then there's a patient control of the same family who does not have bipolar disorder, and made induced pluripotent stem cells. From that stem cells, you make a cerebral organoid. Now this is a 3D structure. You can actually see, I don't know if you can see it very clearly, in the first one there's a leukocyte formation. Okay? There's a leukocyte formation, which is the formation of the neuroectoderm. Okay? Whereas with people, in the patients, what we found was, after 50 days in vitro culture, the vessels did not form. There was no progenitors, proper neuroectoderm structure. It's a living study, the published one, though. I think the publication is not. Okay, fine. So you see a reduced set formation, that means there's a disruption in the neural function. So this is a 3D structure. Again, the power of pluripotency. How you can use pluripotent stem cells to do any kind of basic research. Next. Now, we are speaking about structure here. I made an organoid, I made a 3D organoid, I made a 2D organoid, I found the structure of the cells being disrupted because there's some variant, because there's some disease, right? But then, how do you know if the functions are not done? If the functions are not disrupted? They could be having a variation, there could be a compensation, and the function would not be disrupted, correct? But then, there's a next level. We call them assembloids. So when you look at this, there's something called, so you have neural organoids, means to say, it can be parts of your brain which you make into a 3D structure. But how do they talk with each other? How do we cross-talk with each other? So what we do is put them all together in whatever we want to do. Each stage, if you put it with, it's not here. Yeah. If you put for migration, so I just give you one. So there's the second one, glioblastoma. I think it's not clear what you're projecting.
Next.
Yeah, for you it's clear to me. Okay, that's it. So what I'm trying to say is, different parts of the brain are grown separately. They're made into different cortical organoids. They're made into glioblastoma. For even cancer patients, how to see the interaction between the different parts of your brain, or even with the endothelium, or even with the vascular. So we just grow these different organoids and put them together, and these are called assembloids. Okay, so organoid models, what we do is, then these diseases can give you a defect. For example, I can go to the next study by Sergey Pascal. Here. So what they have done is, they have put two. One, they have put the hindbrain and the spinal cord organoid. They have made an organoid from the hindbrain and they put a spinal cord organoid put together and stimulated it. They did an opportunistic stimulation and they found that user activity in the patients with TCAM receptor reflex is not happening. So this is showing the functionality of the organoids. So we saw the structure, we saw 2D structure, now we are seeing the functionality. Next. I also wanted to put on the fact that these are used, stem cells are not used only for doing basic research, but it's also used in treatment, for therapy. So everybody must have heard it. I'm pretty sure you being biotechnologist, some family members, oh, you're doing biotechnology, do you know stem cells? Are you doing mesenchymal stem cell therapy? What is stem cells? How does it work? I'm pretty sure some of you have heard this question, which is a very common thing. But yes, it is being used for therapy. But as a disclaimer, all the stem cell treatments, none of them are approved across the world. They all are in clinical trial phase. This is something you guys have to spread the news. Do not say that it is approved. If somebody is taking stem cell treatment, please tell them all this in clinical trials. Okay? So, there are n number of clinical trials going on with both adult stem cells and pluripotent stem cells. It's very vast at different diseases, context, pulmonary disease, osteoarthritis, heart disease. Now, but the important context is which stem cell do you use? Do you use the adult stem cell or do you use a pluripotent stem cell? Now that's something which I want to ask the audience. How do I come up with the fact that I should use this particular stem cell for any therapy? Can anybody answer me what is the basis of choosing a stem cell? I told you about pluripotent, I told you about adult. Right? So adult stem cells are bone marrow. From the bone marrow is normal. Bone marrow, umbilical cord blood stem cells, dental pulp. So these all are the sources. But I'm just asking one very simple question. Which stem cells will I take for which disorder? Suppose I say orthopedic disorders. People have arthritis, right? Osteoarthritis. And now in the orthopedic field, it's heavily used. Okay? It's heavily used, the message therapy. What I'm trying to ask is, what is the reason for choosing a particular stem cell? It lies in the basic biology, what I'm asking.
All the cells are integrated in the bone marrow, so associated with the bone.
Okay.
Because of that.
Okay. So I'm asking, okay, I'll ask the other way now. RBC has what life? What's the life of RBC?
120 days.
So, what is the life of your bone cell or osteoblast? Okay, I'll ask the other questions. When is your whole bones, everything replaced in your body? Once in how many years? This is a very basic biology I'm asking. Once in 7 years, your whole bone is replaced. Right? What about your cardiomyocytes? What about your cardiac cells? What about your neurons? When are they replaced? As I told you, stem cells in your body, adult stem cells in your body is for repair, regeneration, regeneration. So how fast will cell is being replaced? That's the basis of choosing your stem cell for repair. Right? So bone marrow is mostly for mesenchymal stem cells and hematopoietic, but hematopoietic is more. That's why in most of the cancers, you use the bone marrow transplant. Okay, yeah, next. So basically, what I'm also getting at, this is now my part of the work. What I'm trying to say is, these all are very complex biological systems. When I say it looks very easy, I grow a cell, no, it's not easy. If I differentiate a pluripotent stem cell, I will not get a pure lineage, which says I have to go to. So what's the next stage from a pluripotent stem cell is the lineage, right? It goes to ectoderm, mesoderm, endoderm, right? A blastocyst becomes a gastroloid. So in the gastroloid, it is the 3 layers: ectoderm, endoderm, mesoderm. What does the ectoderm give rise to? what is the endoderm, and the different tissues. So ectoderm gives rise to most of the neural lineages, right? And the skin. Your mesoderm gives rise to most of the connective tissue in your body. And after that is the endoderm, most of your hormone secretary, your glands. So I'm just giving a few examples. I'm not giving the whole repertoire. I'm just giving you very few examples. What I'm trying to say is that your ecto, meso, endoderm. So when I grow my cells in the dish, if I am pushing it towards the neural lineage, all the cells will not be neural lineage because they have a potential to give you other cell types. So this is where it comes the QC, the standardization, which is extremely difficult. So as in this slide we can put in, so what is important is, so when we want to grow a patient-specific cells, what we do, we collect the patient cells, the somatic cells, make them into induced pluripotent stem cells, with the efficiency is just 0.01%, okay? And then from there, what we do is, we establish certain quality criteria so that it is transplantation ready. What is the criteria for transplantation ready is, one, of course, it should be endotoxin free, it should be mycoplasma free, or free of all the microbes, because it will of course affect the patient. Second is, are they really pluripotent stem cells? Third is it genetically cured? And if I want to do an autologous, there's a student who is trying to cure two cells, two different iPSCs from two different patients. He may mix it up, he or she may mix it up. So then how do I know? That's where the genetics, the STR analysis and the karyotype comes into picture. So karyotype, you say it has a normal karyotype, the STR analysis, the short diagonal repeat analysis will give you an identity of the individual. So it should be pure populations again. Okay, so these are the requirements: identity, purity, viability, sterility, genetic stability, all these are required. If you do not have this, it's like this. And these guidelines are given by the international organization and we have to follow them across the world. It is a unified system. Next. So when I say standardization and resources, how many cells do you require to get into the patient? If I do adult stem cell therapy or pluripotent stem cell therapy, how many cells would I require? You are an adult, your weight is anywhere between 50 to 100 kgs, approximately. I'm just giving an average of all individual adult person. So we say 1 million cells per kg, 0.5 million cells per kg. Again, it depends on the condition. There are a lot of studies to go into it before we do all this. Okay? So, and to even get those cells, a small dish will give you only a million cells. You will require 50, 20 million. And the other factor is when you go into the cells, in the culture, in an artificial condition, you are tending to get genetic alterations in cells. That's a normal thing because it's an artificial environment. So how do you stop that? So again, quality control. We have to do every 10% is always. Okay, so this is a typical quality control pipeline what we do. As I said, that's the first one, verified, that's induced pluripotent stem cell. We say whether it's pluripotent or not by its markers, that is OX4, SSE84, and the TRA180. There are other markers for pluripotency. Then we also show that they can differentiate into any lineage by seeing the qPCR, that is the graph in between. So OX1, OTX, and so they will differentiate them into ectoderm, mesoderm, and endoderm. And then we show. And we use a method called centralization, as I said in the beginning, that when we want to make them into pluripotent stem cells, we put 4 transcription factors into these cells, log4, SOX2, NANOG, to make them into, improve them into pluripotent stem cells. But what is the delivery method? It can be the plasmid, it can be antivirus, it can be adenovirus, it can be just mRNA. But after a few passages, you have to say that the virus is no more present in the cells. So that's another text to do. And then, as I said, a karyotype. Next. Now, people have gone into the next step. Now, you may say that to grow all these cells, maintain these cells, and with high quality control is a really expensive affair. We can't keep doing it from every person. It's nothing but, everybody knows HLE, right? Type 1, type 2. Right? So when you know MNC or type 1, type 2, what do? They have done a rigorous assessment of Indian population, they got blood banks and saw the MNC type 1 and type 2 and found that 20 types are most common in the Indian population and developed GMP grade cell lines so that it can be later used for transplantation so that you don't have any immune rejection. So there's another initiative which is being done in India itself in the part of my institute. Next. So yeah, these are some of the models we developed in our institute. That is HLMI, GMP grade line which I already spoke about. Then we have some stem cell lines developed from healthy donors which are used, extensively by a lot of researchers across India. And then we have developed iPSCs for modeling neurodevelopmental disorders. I will just give you one example of the bipolar disorder. We have for different neurodevelopmental disorders. Then also of modeling blood disorders. The clinical arm of Instinct, CSCR, is developing, has also done the first clinical trial for thalassemia patients, gene-edited stem cell lines, which are put back to patients and has been good. The people are doing greatly in the RP, that's retinal disorders. So there have been a lot of developments, but again, as I say, please do not tell people that it's okay to do stem cell treatment. It's all in the clinical trial stage. Yeah. So this is a stem cell facility which I had. We have a clean room, we have quality control, we do banking to distribution, we make new lines, we also do gene editing, we reprogram on request, and then also we conduct a lot of workshops. I myself in the last 2 years have conducted 5 workshops, and I encourage everybody to apply. Next. So yeah, this is something which I do extensively and I enjoy doing it. Every year I do workshops. Next. So yeah, what is the end point of all this? We are trying to make tailor-made IPSCs, whether for basic research, whether for drug designing, for drug testing, toxicity, or for cell transplantation. We are trying to do that. And yes, this is the method from where you can get, of course, stem cell field is guided ethically. You cannot do stem cell research without consent, ethics consent from the institutional ethics committee or from the even guidance. You cannot touch stem cells. Okay? Yeah. This is the future direction. I have done it all. I have made us employed. I have not made us employed, but I did make good quality IPSCs and made organoids, everything. I'm getting us employed. But you know the one factor which has been, today when you do manual organoids, we get different shape. Each organoid gives us a different endpoint. So automation is the next. So I'm getting to automation. Yeah, and last I would like to thank every person in this journey. Because it's not my effort, it's the whole institute effort. Thank you. Any questions?
Any questions? Please go over for questions.
Thank you very much for your interesting talk. Really, it was fantastic. So I would like to ask one question. So I need one clarity. Yes. So there is a boom for 3D printing technology. So how this stem cell technology can compete with 3D printing technology?
So yeah, you rightly asked. So 3D printing technology is a combination of two things. One, the ECM, the extracellular matrix. Your bone has collagen. It's a hard tissue. Your blood is flowing, your fluid. Then what is your other tissue? Your skin. Any tissue has a different extracellular matrix. So the 3D bioprinting is a mixture of the cells and the extracellular matrix. Without that, you can't make your 3D printing of biological tissues. So yes, it contributes heavily to that.
Madam, are there any cells like stem cells they are using in diagnostic at present? Like for diagnosis and
Right now, not for diagnostic tools, but yeah, to develop diagnostic tools, yes, we are. We're using it. So we use the CRISPR-Cas method, there are many people using it. They use the CRISPR-Cas gene editing method for diagnostics. There are some like that. I mean, it's very vast, the field is extremely vast.
But they are not approved by IVD.
No, that's what I said. Many of it is in the clinical trial stage. It's still nascent, even though it is in the very, 20 years back we introduced the distributed sepsis, because they're very difficult cells to grow.
Madam, one question would be, one question.
Sure.
You are using some inducers to activate the stem cells.
I'm sorry, sir.
You are using some chemicals, inducers, to activate stem cells.
No, sir. What we do is we take the somatic cell. You can take a skin cell, you can take a blood cell, okay? And as you know, the skin cell has the potential to grow on its own. If you put a skin cell in the culture, what happens is you get more skin cells, what we call the fibroblasts, right? You take the blood cells, the progenitor cells, erythroid progenitors or myeloid progenitors, which have the potential to become into another cell type, correct? So these, what we do is we take these kind of progenitors, add to it through plasmid method, plasmids of 4 nanox SOX2. There are 4 transcription factors which drive pluripotency. So we add to that, wait for 28 days, few of the cells pick up and they become pluripotent. They reprogram back.
Is there any possibility of using bioactive molecule?
Yes, there is nowadays people are trying the small molecule method. Not bioactive molecule, but there are some certain small molecules. There are different technologies by which we do. So till now what I described has been, yes, the 4 transcription factors put through different delivery vehicles. Whether you take episomal plasmids, you may take a centivirus, you may take a adenovirus, you may take another method, mRNA or something. But there are small molecules now which are being used for making pluripotent cells. So the driving of a part when we study disease modeling, this is all to study disease modeling and of course come up with some defects. So when we, as I gave you the first example of cardiomyocyte itself, I showed you there is a variant where the cytopenia was disorganized with the cardiomyocyte. When we added stem-wire cycle, what happened was the cytopenia structure got existing. The same way for bipolar disorder, because that is more complex disease. It's not driven by one gene, it's driven by multiple genes. So in bipolar disorder, we are trying to come up what exactly is the mechanism for the person having bipolar disorder. First we study that and then we find out treatment strategy. It drives psychology, but then we have also seen there is the biological entity. Then the set formation is the cortical layer does not form. Yeah.
In US, stem cell therapies became very common in many cases in US. Yeah. So why we can't believe to the other countries? Because we can easily get the stem cells from various resources. So why in Indian perspective is became still unsolved?
Sir, actually US is not a market. Stem cell market is the highest in Thailand, Bangkok and India.
Yeah.
Okay. And these all are unapproved therapies. The other factors also you should understand that these are kind of the other stem cells which they use for this, the autologous stem cells, does not form the cell type which you want. It only goes and gives you the milieu or the environment for the cells to grow. So it is not a proven, that's why I think it's not a proven therapy.
What about CAR T-cell therapy? So it is equal to that?
So CAR T-cell therapy is different, where it's usually used for cancer.
Yeah.
So in cancer, you may have some particular variants. What they do is the CAR T cells, the T cells are made to kill the cancer cells.
Correct.
They identify the cancer cells and kill. So that's the technology and that's very successful.
Yeah.
And it's approved also.
Yeah. Because we can visit the resources abundantly. We can collect it. Why can't we stop it here? We can extend this and we can use it as one of the novel therapy for the individual.
We can use it, but then it won't be quality control, as I've seen on the lab what happens. What I said is that most young cells will behave the way you want it to behave, unless and until you study and give them the right environment.
Okay. Any questions, students, participants? Okay, bye.
Thank you, and thank you for inviting me here. Of course, I've not come back here for a very, very, very long time. I've had my share of challenges and all before I came here. My husband was also a business walked in anyway, so yeah, that's fine. Thank you. I'm proud to stand here, of course.
Chiki sir, please come forward.
So I'm proud to stand here because, of course, the basics comes from here. We are graduating from this department. I don't know about it, we are going to I would like to thank you for inviting me for this talk and congratulating Dr. Kennedy sir for this graduation and hope you have a successful career after that course. Thank you.
Thank you very much. And it is very much insightful and it is very much useful to the young generation to work on these key concepts because it has become one of the newer therapeutic area to work on. So in this extent, we can extend our collaboration with Madam because it is one of the institute, pioneer institute, giving up us a lot of such workshops, or even if you want to work for some of the area of your research with their collaborations, you can easily collaborate and you can utilize this such government facilities. So in that context, I'm very much thankful to Madam also and the organizers also to give up this opportunity to be here. I will give it to the chair sir to continue.
Thank you very much, ma'am, for an excellent presentation on the very important topic, stem cells, how it is useful in the healthcare system today. This is a burning topic as we explained today. Of course, it has not come to the practice field of the medical system, but it has based upon definitely it will come. So we have opened several areas which are very useful to the students and researchers on campus. Definitely, as my colleague said, that we have several areas to collaborate with the departments of biological sciences at the campus. So in the days to come, this will be useful. You mentioned several methodologies. He is involved in, he is conducting several workshops for the students and scientists on stem cell and different factors which are involved, particularly transcription factors which are involved in activating the stem cells. So this definitely talk will be useful to our students. And thank you very much for having presented wonderful talks on this subject.
Thank you, sir. I just want to thank July 1st I'm having a workshop. Last week I'm having a workshop. In September I'm graduating from the conference. We announced it in the beginning of the year, 4-5 workshop, and it's a part and it's a mandate for us. So I would tell all the interested people to please apply for it. It will be eye-opening for you because you will see how things work. The central government does fund a lot in these kind of institutes. You will know what is happening and it will inspire you to research and become greater scientists like us.
Madam, please.
Institute of Stem Cell Science.
Address system NCBS, Bangalore.
Thank you. Thank you.
Now I request Dr. Ganguly ma'am to participate. Speaker Lata Cherikal.
So Malika Chuki is also first best student of our Mumbai Tech, and he also here. clear their